Now let us talk about the urea cycle. The urea is the end product of the protein metabolism. Also, you can say the urea is the end product of the amino acid metabolism. The nitrogen of the amino acid is converted to ammonia, and the ammonia is toxic to the body. The ammonia is eventually converted to the urea and is detoxified. As such, the urea accounts for 80 to 90% of the nitrogen containing substances that is excreted in the urine. The urea is synthesized in the liver and transported to kidney for excretion in the urine. So the amino acid catabolism results in the formation of the common metabolites, that is pyruvate, acetyl coenzyme A. So this pyruvate and acetyl coenzyme A serves as the metabolic fuels. The excess nitrogen generated by this process is converted to urea and is excreted by the kidneys. Now let us talk about the detailed mechanism, the detailed steps how the ammonia is converted to urea. So first reaction of the carbonyl phosphate synthesis and ornithine transcarbamylase, these two enzymes are the mitochondrial enzymes. So this conversion of the ammonia to carbonyl phosphate and conversion of the ornithine to citrulline occurs in the mitochondria. So the ammonia combines with the carbon dioxide, and with the help of the enzyme carbonyl phosphate synthetase 1, the carbonyl phosphate is formed. So this carbonyl phosphate synthetase 1 is the rate limiting enzyme, and the allosteric activator of this enzyme is N-acetyl glutamate. Now after we have carbonyl phosphate, with the help of the ornithine transcarbamylase and the substrate ornithine, the citrulline is formed. So these all the reaction that I have told you, this occurs in the mitochondria. Now from the citrulline, the arginosuccinate is formed with the help of the enzyme arginosuccinate synthase and aspartate, along with the consumption of the ATP. So from the arginosuccinate, arginosuccinase enzyme acts and the arginine is formed. So arginine is formed. So we have the molecule, we have the arginine, and with the help of the enzyme arginase, the ornithine is formed. And while the conversion of the arginine into ornithine with the help of enzyme arginase, and in this step the urea is formed. So the urea has molecule NH2CNH2, that means it has two NH2 groups. The nitrogen of this NH2 comes from the NH3, and this nitrogen of this NH2 comes from the aspartate, and the carbon units of the urea comes from the carbon dioxide that we have here. So we have obtained a molecule urea, and this urea is transported to kidney for the excretion. So this is all about the urea cycle, how urea is produced, and I hope you understood this lecture. So I hope you understood this lecture, and I have illustrated the detailed steps of the urea formation. Again, at last I am going to emphasize on the formation of the urea. So as you can see, the arginase is the fifth and the final enzyme that cleaves the arginase to yield the urea and the ornithine. The ornithine is generated, enters the mitochondria for its rescue in the urea cycle. So arginase is activated by the manganese, and ornithine and lysine compete with the arginine. The arginine is mostly found in the liver, while the rest of the enzyme of the urea cycle are also present in the other tissue. So for this reason, the arginine synthesis may occur to varying degree in many tissue, but only the liver can ultimately produces the urea. So the urea production mainly occurs in the liver. So here, while conversion of the arginine to ornithine, the arginase cleaves the compound arginine to produce the urea and ornithine. So this is the final step. I hope you understood this lecture. So in the following videos, I'll discuss about the transportation of the ammonia by alanine and discuss the various disorder like hyperammonemia and ornithine transcarbamylase deficiency. Thank you.